Well, I'd like to echo Leona's comments about what a pleasure it is to be speaking to this audience on this day, and for that I thank the organizers. And I also have a slide in here in a few minutes which refers to Watson and Crick. So you'll see, I think study of the generation and repair of mismatches during replication of any organism on the Earth, but in this talk, emphasizing work on eukaryotic replication is, for me, what has been my reason for existence as a scientist for the last 40 years. And so what I'd like to do is talk about both how the errors are generated and how they're, during eukaryotic nuclear DNA replication, and how they're repaired primarily by two repair processes, DNA mismatch repair and ribonucleotide excision repair. So these are the determinants of DNA replication fidelity, and they include the control of the substrates used, which means the lesions in the DNA are repaired by a number of different DNA repair processes, which Leona just carefully articulated for you, as well as in the regulation of rNTP concentrations in cells and removing damaged rNTPs before the replication forks operating. At the replication fork itself, there is the nucleotide selectivity of DNA polymerases and proofreading exonuclease. Occasionally, these mechanisms don't operate so well, and so forks stall as these two processes attempt to accurately replicate the DNA, and that results in stalled forks, and there are a number of repair processes that DNA transactions that are in cells for resolving those stalled forks. And then if replication is successful, it occasionally leaves behind mismatches or newly incorporated ribonucleotides in the DNA. So what I want to talk about in my talk is the role of nucleotide selectivity, proofreading, and post-replication repair processes on determining the fidelity of eukaryotic nuclear DNA replication. So these are the catalytic subunits of the known DNA polymerases in humans and E. coli. So pol 1, pol 2, pol 3, pols 4 and 5 are in separate families, and this is the replicative polymerase, and the other ends are the major replicative polymerase for undamaged DNA. And the other polymerases have more specialized functions. And so too, eukaryotes are now known to encode 17 different DNA polymerases, and they're given designations of the Greek alphabet. The first one discovered was pol alpha, and so on. And they can be classified into classes of polymerases with homology, and the three that I want to spend the remainder of this talk on are alpha, delta, and epsilon, alpha, delta, and epsilon polymerases. And from these colors, you see that only a small number of DNA polymerases have the yellow code, which is for a proofreading exonuclease. So the majority of eukaryotic DNA polymerases, or the majority of polymerases in general that we know about, lack the ability to proofread errors. This is the fidelity of the eukaryotic DNA polymerases measured in vitro by a number of investigators. And in this designation down here are the proofreading deficient and the proofreading proficient DNA polymerases from mammalian or yeast cells. And you can see that the proofreading proficient DNA polymerases are the most accurate on this sliding scale from no discrimination against errors to a fidelity in vitro that is at least as good as 10 to the minus 10 errors per nucleotide replicated, and that comes from nucleotide selectivity, proofreading, and mismatch repair, And in vitro, the nucleotide selectivity and the proofreading capacity of polymerases varies widely from experiments published by us and others on DNA polymerase epsilon, which can almost make one mistake per nucleotide polymerized under some circumstances, to the accurate DNA polymerases, pol gammas for the mitochondrial replicase, and pols delta and epsilon are the proofreading proficient eukaryotic replicases, and they're quite accurate. And there's a lot of variation in these numbers depending on the details that we and other experts in the field have elucidated over time. What I'd like to do is give you just a small taste of a large amount of information that we and others have discovered about the binding pocket for the nascent base pair of accurate DNA polymerases, that is, those with high nucleotide selectivity. This is the X-ray crystal structure of one of the replicases, and this is a newly forming base pair with dideoxy ATP incoming opposite a template T. And you can see that the primer terminal base pair and the O helix of this enzyme form a very tight binding pocket as a result of both DNA and dNTP binding. This pocket closes on this nascent base pair. It excludes water and checks base pairing geometry. Correct base pairs fit this pocket snugly and trigger polymerization. Incorrect base pairs with non-canonical geometry reduce the rate of chemical bond formation, and accurate polymerases, especially the eukaryotic replicases, check geometry via polymerase interactions with the DNA minor groove. So that's a quick way to describe a tremendous amount of valuable information provided by X-ray crystallographers and biochemists over the years. If the polymerase makes a mistake, it can be proofread by the exonuclease activity associated with a replicase. That's some 30 angstroms away from that active site I just showed you. And that's one major error correction mechanism for a variety of organisms, including humans. This is the final one, which is eukaryotic DNA mismatch repair. This is the pathway that Paul Modrich won the Nobel Prize for mechanistic studies of for the last 30 years. It's brilliant work. It starts with mut-S alpha recognition of a mismatch. This protein is a heterodimer of two homologs of the bacterial mut-S protein, which gets back to what Leona said in her talk, that most of the repair pathways we know about were first discovered in prokaryotes, and it wasn't until 10 years after mut-S alpha in bacteria was discovered that its homologue in eukaryotes was discovered. This recognizes the mismatch, and that's followed by binding of mut-L alpha, a second protein comprised of PMS2 and MLH1 in humans. And after this binding, a miracle occurs. We're still trying to figure that out, but it signals to tell the cell which strand contains the mismatch. In this case, it's the green strand, which represents the newly replicated strand. It's essential for mismatch repair to recognize the error in the newly replicated DNA as compared to the parental DNA. And that leads to strand incisions, which then leads to mismatch removal by any one of three different mechanisms. And then after the mismatch has been removed, it leads to DNA resynthesis. We certainly know that pol delta can operate in that regard, and possibly pol epsilon. And then once the DNA has been resynthesized and you ligate, you've fixed the mistake up here. So you could give a whole Cold Spring Harbor symposium, week-long series of talks on just mismatch repair. It's so well known, and for those of you who are interested in the details of this process, within the last month or so, an issue of the journal DNA Repair came out that was totally devoted to a whole lot of different subjects related to mismatch repair. So it is a very widely interesting repair pathway. And as I said, much of what's on this slide was defined by Paul Modrich, for which he won the Nobel Prize in December. The second kind of error is ribonucleotides. Are they incorporated during DNA replication? This is a fidelity issue, but for the sugar, for an incorrect sugar, not an incorrect base. And one example of why we are interested in this, my lab is interested in the fidelity of DNA replication. And so we got interested because if there's a 2' hydroxyl of a ribose in a DNA molecule, it can attack the sugar phosphate backbone and cleave it to leave a cyclic 2', 3' phosphate and a 5' hydroxyl, which need to be processed in order to be repaired correctly. And in that processing is the opportunity for aberrant chemistry to take place and therefore mutations to arise. So since that's been realized for a long time, there's a whole body of work, especially by the crystallographers and biochemists, showing that there's a particular tyrosine residue in a replicase that if there were a 2' oxygen on this, a ribose ring of an incoming triphosphate, that is prevented from being incorporated by that tyrosine residue and residues in the local vicinity. So there's a lot of really good reductionist biochemistry done on how polymerases exclude ribose, but what they didn't take into account, and we did in 2009, was the fact that the DNTP and the rNTP concentrations in cells, be they yeast or human cells, is highly imbalanced. So this is the micromolar concentration in vivo of the 4 deoxynucleotides for polymerization, and this is for the ribonucleotides for all the RNA transactions that occur in cells. And so the numbers vary, but there's about 100 times more ribose than deoxyribose in cells. So we got interested in whether they're incorporated in vitro. This is the actual assay that we used for that. And this is the result with DNA polymerase delta, DNA polymerase alpha, and DNA polymerase epsilon. And these bands here and these gels represent locations where in a simple in vitro polymerization reaction, these wild-type yeast DNA polymerases readily incorporated quite a few ribonucleotides into DNA. And that really shocked us because the first time I saw this result, we did a calculation and we came up with the following logic. This was a table put together by Thomas Lindow, who was also one of the Nobel laureates for his work on base excision repair. He put this together in the early 1900s and gave it to me when I wrote a survey, a review of a meeting that we had both attended. And it lists the number of lesions created in a human cell, in the human genome in a 24-hour period, and those numbers are here. And there's been 30, 40 years worth of work on DNA repair of these kinds of lesions and the consequences of failure of that repair. If we give those numbers little dots, where this littlest dot might be hypoxanthine, for example, and the biggest dot might be an abasic site, this is what we see for ribonucleotides. Ribonucleotides are incorporated into DNA by wild-type replicases at somewhere between 100 and 1,000 times more frequently than all of the other lesions that had been studied for the last 40 years. So how are they misincorporated? If they are misincorporated, how are they repaired? And if they aren't repaired, what are the genetic consequences becomes a very interesting question that became of interest to quite a few people starting in 2010 when we published this result. So now I want to focus on the next part of the talk, which is in duplex DNA, the two strands are oriented anti-parallel, as Watson and Crick taught us in 1953. DNA polymerases copy templates in only one direction, and that was taught by Arthur Kornberg et al., first in a BBA article published in 1956. Since then, lots of polymerases have been discovered, and they all conform to this statement. Therefore, replication of duplex DNA is intrinsically an asymmetric process because the two strands are anti-parallel, but a polymerase can only copy in one direction. And we, as a subset of many questions that this brings up, are interested in what is the relationship, if any, between DNA replication asymmetry and DNA replication fidelity. So we've been studying that for most of my career, and I'd like to give you, for the rest of this talk, some of the information that we and many others have gained. These are the two major eukaryotic nuclear replicases, Pol delta. In yeast, it's a 3-subunit polymerase. In humans, there's a 4th subunit. And this is Pol epsilon. And these two enzymes are the two most accurate DNA eukaryotic replicases among the 17 that I mentioned that I know about. And these are some of their properties, but they're not identical properties. And therefore, it becomes interesting if you're interested in whether asymmetric replication has consequences for the fidelity of replicating the Watson and the Crick strands, which are oriented anti-parallel, whether these functions of these, what are the functions of these two DNA polymerases. So we can summarize where the field is right now by this beautiful review written by your president, Bruce Stillman, and published a few months ago in Molecular Cell, in which one model for which of these two polymerases replicates which strand would be that Pol delta primarily replicates both strands. And that highly accurate DNA polymerase replicating both strands was what Bruce and his postdoc demonstrated in 27 years ago now, for the SV40 virus. That model is still popular in some minds, with the later knowledge that a third polymerase might engage in DNA replication and could occasionally be used. So under normal circumstances in yeast, for example, a model was put out which stimulated this publication, which states that Pol delta replicates both strands, and when Pol delta occasionally makes a mistake that can't be proofread so well, Pol epsilon, which is the third polymerase at the fork, can occasionally take over and proofread an error made by Pol delta. And you could rationalize the involvement of this third polymerase as being involved in cell chromosome replication following DNA damage. Or you could have a model in which Pol delta is the major lagging strand replicase and Pol epsilon is the major leading strand replicase. And I will prejudice you by saying that the rest of my talk is derived from this model for the replication of undamaged normal DNA. So that's what I'm going to talk about from now on, is how is undamaged normal eukaryotic nuclear genome replicated and which polymerase operates on which strand. And that does not deny any of these models yet. In fact, the model that we have is just that, a model. I'm a firm believer in it's difficult, if not impossible, to prove anything in science, so we just gather information that is consistent or not with any particular model. So this is our favorite hypothesis right now for the normal replication enzymology of the eukaryotic nuclear DNA replication fork. So one reason we believe that is, and this is probably the strongest reason we believe that, is we developed a procedure called hydrolytic DNA end sequencing, which took advantage of the fact that replicases incorporate ribonucleotides into DNA, and we developed a high throughput screening method to ask, given that ribonucleotides render DNA sensitive to treatment with alkaline conditions. In this kind of procedure, we can ask in strains that are defective in the ability to repair ribonucleotides incorporated by each of the three replicases, alpha, delta, and epsilon, where are DNA ends generated in our case in budding yeast. And this is the answer: HydEn-Seq mapping of ribonucleotides in ribonucleotide excision repair deficient yeast. This is just one tiny piece of chromosome 10. We have the information for the whole genome. But what you can see is the blue lines represent the density of DNA ends in RER-deficient cells treated by the HydEn-Seq procedure when the DNA is extracted from a yeast cell containing a mutant derivative of pol epsilon that incorporates a very large number of ribos. And you can see that the ribonucleotide density is essentially strand-specific. So in this case, there's very few ends on this side of this origin of replication on chromosome 10, but there's a large number on this side. And so if you stare at this kind of information with this kind of replication enzymology depicted, you realize from this kind of information that, or you reach the conclusion that ribonucleotides in a pol epsilon strain are primarily on the leading strand, and ribonucleotides incorporated by a mutator derivative of pol delta or alpha are primarily on the lagging strand, and the switch occurs at origins. So this is consistent with this model where the leading strand is primarily replicated by pol epsilon and the lagging strand is primarily replicated by pols alpha and delta. Now there are other interpretations to this if you like one of the other models better, as did the Prakash in their molecular cell paper about 8 months ago. So if you are interested enough in this topic, you can read their point of view on this. But this leads us to the model where pol epsilon is the major leading strand replicase and pols alpha and delta are the major lagging strand replicases for normal undamaged DNA. This is the ribonucleotide excision repair pathway. And so that we put the ribonucleotide on the leading strand, and the repair pathway involves incision by RNase H2, followed by strand displacement synthesis, primarily by pol delta, but pol epsilon can also work to some extent in an in vitro reaction. And once this strand displacement occurs, flap processing removes this ribonucleotide, allowing ligation to correct the ribonucleotide incorporation event. This was the in vitro part of this was primarily done by Peter Bergers, our collaborator on much of this work. And then there are some alternatives. Anytime you have a repair pathway, if it fails and can screw up a replication fidelity. So one of those is a process in which RNase H2 acts to generate a ribonucleotide, and when the ligase attempts to ligate this off-pathway transaction, then the ligase can, in its normal mode, add an AMP residue to this end, but since it's linked to a ribonucleotide, that's a problem that's usually dealt with by an enzyme called apritaxin. But if that enzyme is deficient in mice or humans or in yeast where we work, then you get slow growth, replication stress, and genome instability, and that's associated with a disease called a Cardi-Gutierrez syndrome. An alternative pathway aberration would be inactivation of RNase H2, which leads to resident ribonucleotides in the genome, which can be incised by topoisomerase 1. Topoisomerase 1 incision of ribonucleotides creates a 5' hydroxyl and a cyclic 2', 3' phosphate. Those have to be repaired, but in so doing, you get replication stress and genome instability. So these are alternative problematic pathways to aberrations in ribonucleotide excision repair. This is a summary slide of what we know about the origins, the repair, and the consequences of ribonucleotides in the genome. And I'm not going to try to go through all of that, but if you're interested, Jess Williams and I wrote an article for for DNA Repair in 2014, and for Nature Reviews Structural Molecular Biology that is just coming out. And it encompasses all of this kind of information. And I should also point out that some of these phenotypes may have to do with roles of the RNase H2 enzyme in incising ribose that have nothing to do with ribose incorporated during replication, but could be involved in resolving R loops that arise during transcription, for example. So this part of the process is highly complicated. Let me go back and show you positive consequences. We think there's a chance that ribonucleotide incision by RNase H2 is an important strand discrimination signal for mismatch repair, and that's illustrated here. This is motif A of the eukaryotic replicases, where there's a leucine or a leucine that we've been manipulating in the lagging strand replicases, and that is conserved as a methionine in pol epsilon, which we believe is the leading strand replicase. And if you do the reductionist biochemistry, ribo incorporation compared to deoxyribo incorporation for the wild type enzymes is these values, but for the mutant enzymes that we've been investigating, they're these values. And you can see that methionine in pol epsilon is less selective against ribose than is leucine. And pol epsilon is one residue for more efficient ribo prevention. This amino acid is conserved in pol epsilons, and our data suggests that pol epsilon primarily replicates the leading strand, and therefore we wondered whether this is a methionine and these guys are leucines because this provides some positive selective advantage to an organism. And we speculated on mismatch repair. So suppose that the polymerase makes a GT mismatch, but it also occasionally incorporates a ribonucleotide, that RNase H2 could incise this ribonucleotide to provide a strand discrimination signal for removal of the mismatch. So another way to state that is in a review that Dorothy Erie and I wrote on strand discrimination during eukaryotic DNA mismatch repair that came out a few months ago. So the main strand discrimination signal for mismatch repair, as I articulated before, is an incision by mut L alpha. A second one would be involving repair of pol alpha errors, but a third one for leading strand replicase errors could be proofreading or processing ends generated by RNase H2. And that could be a minor component of the mismatch repair signal in eukaryotes. At least all the data that we and Joe Jurikny's lab have generated are consistent with that hypothesis. There may be other possible positive consequences. We're not experts in any of these subjects, but we posit that possibly RNase H2 incision of ribonucleotides might be one reason why these processes exhibit the specificity that they do. And that, like any relatively recent field, and the consequences of ribose in DNA are largely unexplored. There are lots of possibilities for future work, especially for you postdocs in the lab who are interested in this. We described whole genome mutation rate analysis of 8 diploid yeast strains, and that was published in 2014. And it basically takes advantage of wild-type polymerase, either with or without mismatch repair, or the pol epsilon strains that I've been alluding to that have a methionine to a glycine, a leucine to a methionine, a leucine to methionine, plus or minus mismatch repair. And we designed a whole genome sequencing protocol, being now fans of whole genome sequencing, to ask where mismatches are placed in each of these 8 yeast strains. And the answer is here. These are the data on the number and distribution of single base changes introduced into a cell that has all wild-type polymerases but is mismatch repair defective or proficient, a pol alpha variant that is a mutator enzyme, a pol delta variant, and a pol epsilon variant. So if you're interested in this, that work is published, but I'm going to show you a taste of the data right now. This is the rate of generating replication errors, and these are results in mismatch repair defective strains. So these are only able to take advantage of nucleotide selectivity and proofreading, not post-replication mismatch repair. And in a pol epsilon plus strain, the mutation rate per genome is 0.38. That means that only 1 out of every 2.5 yeast cells even has a single base substitution anywhere in its genome. In other words, replication fidelity depending on nucleotide selectivity and proofreading from this estimate of whole genome mutation rates is incredibly accurate process. And this is the results for the mutator alleles, which generate a lot more errors in the genome, in fact, this number. So let me just talk about this work first in mismatch repair defective strains. You can map where the mutations are across the genome, 5,164 base substitutions that were AT to GC transition mutations. And knowing that this derivative of pol delta puts errors, makes GT mismatches about 20-fold more often than it makes the reciprocal CA mismatch on this strand. You can map these to origins of replication, and you get a pattern that looks like this, where this is many of those mutations at one origin and very few at the next origin, where this is the right side of the origin and the left side of the origin. And this is the reverse for this TG mismatch over here. And if you do enough of that kind of stuff, this is the result I just showed you, and this is for all 12 mismatches. All of these patterns, to put it bluntly, are consistent with the model in which pol epsilon is the primary leading strand replicase and pol delta is the primary lagging strand replicase for the normal nuclear genome with undamaged DNA. And moreover, if you then ask what happens in a cell with all wild-type replicases, although there's many fewer mutations, they conform to the pattern consistent with pol alpha and pol delta mutations. So we conclude from this, tentatively, that lagging strand replication in the absence of mismatch repair is about 2-fold less accurate than leading strand replication. We are very interested in what determines whether a mutation arises. And so we've mapped what happens within 10% of these origins. And it leads to a pattern of misincorporation in this whole genome sequencing analysis, which involved the analysis of 40,000 single base SNPs. That base stacking promotes infidelity by stabilizing mismatches and promoting their extension to reduce proofreading. And beyond this statement, there are a number of other statements that are possible from this publication. When mismatch repair is absent, error rates vary with these variables for reasons that we can speculate about and are investigating. This is the mutation rate of replication errors in mismatch repair proficient or deficient cells. This is the equivalent mutation rate if we leave mismatch repair intact. And the difference in these mutation rates per genome are these values, which are the average contributions of mismatch repair to replication fidelity. And there's one important concept here, and that is that errors made by pols alpha and pol delta are repaired approximately twofold more efficiently than errors made by pol epsilon. And so while in the absence of mismatch repair, replication fidelity is less accurate for lagging strand replication, mismatch repair is more efficient. Therefore, obtaining as a consequence of all three fidelity steps, the same fidelity of replication of undamaged DNA on both the leading and the lagging strands. And these are some of the mismatch repair efficiency values, statements that we can make from this work, that variables that are or may be relevant to 100,000-fold variation in mismatch repair efficiency in yeast include the ones in black as demonstrated by our work and as speculated by some of our work, these ones that need a lot more further experimentation. And then this is the very last point. What I've just described to you partly is an analysis of strains that are deficient in these processes by virtue of using mutator derivatives of pol alpha, delta, and epsilon that have mutations in the polymerase active site. But those are artificial mutations, whereas if we make mutations in the exonuclease active site that inactivate the proofreading exonuclease of the enzyme, then we're more akin to what might happen in a cancer cell, for example. So we've been doing that, except that so far we've only had time to do that in strains in which we've looked at mutation rates at a specific reporter gene that represents 1/10,000th of the yeast genome. And these are the results of that, which were published a few months ago, in which these are the mutation rates of these strains, and these are the nucleotide selectivity, proofreading, and mismatch repair contributions determined for the leading strand replicase and the lagging strand replicase. And from this kind of looking under the lamppost examination of URA3 gene, we can make a couple of statements. In vivo, the nucleotide selectivity of what we think is primarily pol epsilon errors is this value. But in vitro, we have years of work demonstrating that in vitro, the polymerase pol epsilon has a nucleotide selectivity of 10 to the minus fifth to 10 to the minus sixth. And that's a 3,000-fold difference. And we also know that we also infer that on the leading strand, proofreading is less efficient than on the lagging strand, and mismatch repair is also somewhat less efficient than on the lagging strand. So those statements are here, and that leads to the question are these phenomena 3,000-fold higher nucleotide selectivity in a cell than in vitro really true for the whole genome? And if so, why is that the case? So that's one of the major questions that Zigong Zhu in my lab is currently investigating for the whole genome to see if we can recapitulate or not this kind of information done at URA3. So this is the summary slide. I'm going to stop here, but just remind you that the error rates in vitro of the polymerases that I've talked about are here. The ribonucleotide incorporation propensities are here. This is mismatch repair, and this is ribonucleotide excision repair. And we're very interested in the relationships of these three major processes to cells. And the last thing to say is defects in all three of these processes are very strongly associated with human diseases. So I was very pleased last night when I got into my room and was reading the little Cold Spring Harbor brochure and reading the comments put forth by your president of the value of basic research. What I've tried to do is describe to you the value of basic research in a yeast model organism that definitely has a lot of value to understanding the origins of human cancer or neurodegenerative diseases. Thanks for your attention.
forgot to mention that Tom does work at the National Institute of Environmental Health Sciences. Questions?
If Pol epsilon is mutated, will another polymerase fill in for it? And if so, is it as efficient or less?
Yeah, that's one of, so the answer is yes. You can completely inactivate the polymerization activity of Pol epsilon and get a viable yeast cell. There are 4 really beautiful publications on this from 1999 through 2001. And this is part of the rationale which justifies the conclusion in support of the Prakash model that Pol epsilon is not essential for leading strand replication. Pol delta does it, but it can contribute to fidelity by proofreading. So yes, that's the strongest argument in favor of the Prakash model for proofreading by Pol epsilon. We do not agree with that conclusion at all for reasons that are described briefly in a response in Molecular Cell to their article. Saying work that isn't published yet, but we're really working hard on it, is those 4 papers that I described are fantastic pieces of work, but they all miss the point that when you inactivate Pol epsilon, you do get survivors, but they're so sick it's close to death. And so what we're talking about is normal replication of undamaged DNA. And so we still favor our hypothesis.
I'll just add that Tom and I both know that not only are those Pol epsilon mutants sick, they all carry a suppressor. And we're trying to map the suppressors, both of us.
Hi. I wonder if you would like to reveal more about the replication and polymerase usage in relation to other chromatin events like transcription, like you briefly talked about R loops and other chromosome density in these.
Yeah, so the best thing to point out in that regard is work by Sue Jenks Robertson and Thomas Pettis, who just published a paper which strongly argues that in addition to the phenomena I talked about, ribonucleotide incision is highly relevant to the resolution of R loops, and when that fails, you get strong mutator effects for deletions of 2 to 5 base pairs in repeat sequence elements, and you get larger forms of genome instability. So I'm glad you asked that question, because that statement was put in there, that slide, intentionally to tell you that if you're interested in this topic, you need to be looking at a whole bunch of papers that we have nothing to do with, that investigate a completely separate process. Well, that investigate transcription, and I don't think transcription and replication are necessarily independent of one another, so it's not really a completely separate process, but there is plenty of room for arguing in existing literature by others that in addition to what I've emphasized here, RNase H2 effects on ribonucleotide incorporation influence genome stability through resolution of R loops and retrotransposons.